Bismillahirrahmanirrahim. In this lecture, we will discuss about the gram-negative cocci, that is the Neisseria gonorrhea. Neisseria is a genus given the name because of the scientist Albert Neisseria, and the gonorrhea literally means the flow of the seed. Gonorrhea is the sexually transmitted disease. It is the second leading cause of the sexually transmitted disease after the chlamydia. General characteristics of the Neisseria gonorrhea. Neisseria gonorrhea, it is a gram-negative cocci, mainly found in the kidney shaft, occurs in pairs. That's why it is a diplococci, having no flagella, non-motile, and having no H antigen. Neisseria gonorrhea is non-spore forming, having no capsule, so non-capsulated in comparison with the Neisseria meningitis, having the capsule. Neisseria gonorrhea only ferment the glucose, while the meningitis ferment both glucose and maltose as well. Facultative anaerobes. As we have discussed that Neisseria gonorrhea, that is also called the gonococcus, has no polysaccharide capsule but has multiple serotypes based on the antigenicity of its pilus protein. Its pilus protein has a variety of arrangements. That's why it is called the heterogeneous bacteria, heterogeneous infection, because of the variety or because of the phase variation in the pilus. There is a marked antigenic variation in the gonococcal pili as a result of chromosomal rearrangement. More than 100 serotypes are known because of these uh, rearrangement that are mainly used in the pili protein. Gonococci have three outer membrane proteins: protein 1, protein 2, and 3. Protein 2 plays a role in attachment of the organism to cells and varies antigenically as well. प्रोटीन 2 का जो रोल है वो अटैचमेंट में और ये प्रोटीन 2 एंटीजेनिकली वेरी करती है बिकॉज ऑफ द वैरायटी ऑफ द प्रोटीन 2 in different gonorrhea. Neisseria are gram-negative bacteria and contain the endotoxin in their outer membrane. As it's a gram-negative, so gram-negative bacteria mainly produce the endotoxin. That is the part of its outer membrane, and that is the lipopolysaccharide. In case of the Neisseria gonorrhea, the polysaccharide is not elongated, it is a short. That's why it is the lipooligosaccharide instead of the lipopolysaccharide. The endotoxin of the Neisseria consists of lipooligosaccharide, in contrast to the lipopolysaccharide found in enteric gram-negative rods. जो अदर बैक्टीरिया हैं उन सब में लिपोपोलिसैकेराइड होता है, लेकिन नाइजेरिया गोनोरिया में जो एंडोटॉक्सिन है वो लिपोओलिगोसैकेराइड है. That's the lipooligosaccharide. Both lipopolysaccharide and lipooligosaccharide contain lipid, but the oligosaccharide part of the lipooligosaccharide contain few sugars. इसमें जो carbohydrate chain होता है, जो sugar होते हैं, वो short होते हैं as compared to the polysaccharide chain. Whereas the polysaccharide part of the lipopolysaccharide contain long repeating sugar side chains. तो मेजर डिफरेंस क्या है लिपोओलिगोसैकेराइड में और लिपोपोलिसैकेराइड में? लिपोओलिगोसैकेराइड में जो शुगर चेन होता है वो शॉर्ट होता है, जबकि लिपोपोलिसैकेराइड में शुगर चेन लॉन्ग होता है. The growth is inhibited by toxic trace metals and fatty acids found in certain culture media, mainly blood agar plates. Blood agar plates में जो growth है वो inhibit होती है because it produce some type of chemicals that inhibit the growth in the blood agar. They are therefore cultured on the chocolate agar and containing blood heated to 80 degrees centigrade, which inactivates the inhibitors. That's why we choose a chocolate agar for the growth of the Neisseria gonorrhea. Neisseria are oxidase positive. They possess the enzyme cytochrome C. This is an important laboratory diagnostic test in which colonies exposed to phenylene diamine turn purple or black as a result of oxidation of the reagent by the enzyme. फिनाइलिन डाइअमाइन को जो कॉलोनीज हैं वो उसको पर्पल कर देंगी. द पर्पल कलर शोज दैट द ऑक्सीडेज पॉजिटिव एंड नाइजेरिया इज ऑक्सीडेज पॉजिटिव. Both Neisseria gonorrhea and meningitis both are the oxidase positive. Influence factors of the Neisseria gonorrhea. Neisseria gonorrhea is antigenically heterogeneous. Heterogeneous means a variety of antigens are found in the Neisseria gonorrhea and capable of changing its surface structure in vitro and presumably in vivo to avoid host defenses. It can change its structure to avoid the host defenses. Surface structure include the following: pili. Pili are the hair-like appendages that extend up to several micrometers from the gonococcal surface. Gonococcal surface से जो hair-like appendages extend होती हैं, that are called the pili. Pili mainly mediate the exchange of genetic material between strands and also help in the attachment to human mucosal cell surface, invasion of host cells, and survival through the inhibition of phagocytosis. पिलाई का मेजर रोल एक्सचेंज ऑफ जेनेटिक मटेरियल है एंड सेकंड इंपॉर्टेंट रोल अटैचमेंट टू द म्यूकोसल सरफेस है एंड invasion of the host cells, and it also help in the survival of the cell Neisseria gonorrhea in the phagocytic cells. Two types of the pili, type 1 to type 2 virulent, and the type 3 and to type 5 are the avirulent. Genetic phase variation of pillar structure between type 1 through type 5 allows the organism to vary its antigenic structure, preventing ignition by host immune cells. Chunki a variety of proteins are available, that's why there are antigenically different types of bacteria are present that can use in the prevention of the in the evasion of the immune cells. Porin protein, that is called the pore proteins. Pore protein extends through the gonococcal cell membrane. Cell membrane से extend करती हैं pore proteins. It form pores in the surface through which some nutrients enter the cells. Pore protein mainly responsible for the formation of pores in the membrane that allows the movement of or the entry of the nutrients into the cell. Pore proteins may impact intracellular killing of gonococci within the neutrophils by preventing phagosome-lysosome fusions. पोर प्रोटीन का सेकंड इंपॉर्टेंट रोल क्या है कि पोर प्रोटीन फेगोसोम लाइसोसोम फ्यूजन को इनहिबिट करती हैं. जब फेगोसोम लाइसोसोम फ्यूजन नहीं होगा तो प्रोसेस ऑफ फेगोसाइटोसिस इनहिबिट होगा. सो पोर प्रोटीन आल्सो हेल्प इन द इवेजन ऑफ द Neisseria gonorrhea by the process of the phagocytosis, evade the process of phagocytosis by preventing the phagosome-lysosome fusion. In addition, variable resistance to gonococci to killing by normal human serum depends on whether pore protein selectively binds to complement components C3b and C4b. Opacity protein, that is also called the OPA protein. These proteins function in adhesion of gonococci within colonies and in attachment of gonococci to host cell receptors such as heparin-related compound and CD66 or carcinoembryonic antigen-related cell adhesion molecules. The OPA protein are responsible for the attachment or the adhesion of gonococci. Reduction modifiable protein, that is called RMP protein. RMP, reduction modifiable protein. It is antigenically conserved in all gonococci. It associates with the pore in the formation of pores in the cell surface. Pore protein के साथ association में pores बनाने में helpful होती हैं RMP protein. It blocks the bactericellular effect of host IgG, जो immunoglobulin G है उससे प्रिवेंट करती है, host resistance प्रोवाइड करती है, बैक्टीरियल रेजिस्टेंस प्रोवाइड करती है. Lipo-oligosaccharide. Gonococci lipopolysaccharide does not have long O antigen side chain and is called the lipo-oligosaccharide. As we discussed, इसका जो sugar moiety है, sugar contents है वो short है, that's why it is called the lipo-oligosaccharide instead of the lipopolysaccharide. It acts as an endotoxin. Toxicity in gonococcal infection is largely attributable to the endotoxic effects of lipo-oligosaccharide. Lipo-oligosaccharide, endotoxin, and outer cell membrane protein 1 to 3 are important in antigenic variation and for eliciting an inflammatory response. Lipo-oligosaccharide that act as endotoxin and the outer cell membrane protein, जिसमें protein 1 to 3 हैं, these proteins are important in the antigenic variations and for eliciting an inflammatory response. Other proteins that act as an antigen are valence factors. H8 is a surface exposed protein that is heat modifiable like OPA. The FBP, ferric binding protein, similar in molecular weight to pore is expressed when the available iron supply is limited, such as in human infection. Gonococci elaborate an IgA1 protease that splits and inactivates IgA1, a major mucosal immunoglobulin in humans. IgA1 protease that inhibit the IgA1 antibodies, and it help in the entry and it help in the prevention of the antibodies. जो IgA है that is a secretory antibody mainly help in the mucosa. अगर secretory antibody को डिटॉक्सिफाई कर लेंगे IgA protease या उसको सेपरेट कर देंगे इनएक्टिवेट कर देंगे then it will help in the pathogenesis of the gonococci. Summary of the valence factors of Neisseria gonorrhea. The most important valence factors include the pili, that is the extension of the cell, lipo-oligosaccharide mainly present in the outer membrane, porin proteins, opacity proteins, and the part of the outer membrane act as valence factors that help in the pathogenesis of the Neisseria gonorrhea. Pathogenesis and epidemiology of Neisseria gonorrhea. Gonococci, like meningococci, cause disease only in humans. The reservoir is the human, and the infection is only caused in the humans. There is no other reservoir other than the human. The organism is usually transmitted sexually. Newborns can be infected during birth, as it is a sexual transmitted disease, so it transmits mainly sexually, and the newborns, the neonates, can infect during the birth. Because gonococcus is quite sensitive to dehydration and cool conditions, sexual transmission favors its survival. तो gonococcus से ये dehydration और cool condition से sensitive है, kill हो जाता है, इसलिए mainly survive in the moist condition. Gonorrhea is usually symptomatic in men but often asymptomatic in women. Genital tract infections are the most common source of the organism, but anorectal and pharyngeal infections are important sources as well. Along with the genital tract infection, it also causes the pharyngitis, it also causes the anorectal infections and other body infections. The main host defenses against gonococci are antibodies IgA, secretory antibodies, and IgG, the chronic cases, complement, and the neutrophils. Antibody-mediated opsonization and killing within phagocytes occur, but repeated gonococcal infections are common, primarily as a result of antigenic changes of pili and outer membrane protein. Outer membrane protein और villi चूंकि अपनी जो चेंज जो स्ट्रक्चर है उसको चेंज करते रहते हैं, इसलिए इसका जो इसकी वैक्सीन भी अवेलेबल नहीं है और इसकी पैथोजेनेसिस बहुत स्ट्रांग होती है. सम टाइम द बैक्टीरिया हैविंग द पिलाई ऑफ वन टाइप कैन इन्फेक्ट एंड द अदर टाइप बैक्टीरिया हैविंग द पिलाई ऑफ द अदर टाइप इन्फेक्ट, सो immunity cannot help in the eradication of all types of the gonorrhea. Gonococci infect primarily the mucosal surfaces, the urethra and female genital tract, but dissemination can also occur. Dissemination means the spread to other organs as well. Certain strains of gonococci cause disseminatory infections more frequently than others. The most important features of these strains is their resistance to being killed by antibodies and complement. The mechanism of serum resistance is uncertain, but the presence of the porin protein porin A in the cell wall, which inactivates the C3b component of complement, appears to play an important role. The occurrence of a disseminatory infection is a function not only of the strain of gonococcus but also the effectiveness of the host defenses. Person with a deficiency of the late-acting complement component, that is C6 to C9, are at risk for disseminated infections or women during menses and the pregnancy or gestation period. वो पर्सन जिसमें C6 to C9 जो जिसको हम लेट एक्टिंग कम्प्लीमेंट प्रोटीन कहते हैं वो डिफिशिएंस होंगे, उसमें प्रॉब्लम होगा तो उसमें डिसेंट्री इन्फेक्शन के चांसेस ज़्यादा होते हैं. Mainly it occurs in the women during the reproductive cycle and the gestation period. Dysentery infections usually arise from the asymptomatic infections, indicating that local inflammation may determine the dissemination. Clinical finding of the gonococcal infections. Gonococci cause both localized infection, usually in the genital tract, and disseminated infections with seeding of various organs. Gonococci reach these organs via the bloodstream gonococcal bacteremia. Gonorrhea in men is characterized primarily by urethritis, inflammation of the urethra accompanied by dysuria, and a purulent discharge through the urethra, yellowish color of the discharge, and inflammation of the epididymis called the epididymitis may also occur. In women, infection is located primarily in the endocervix, causing a purulent vaginal discharge and intermenstrual bleeding that is called cervicitis. The most frequent complication in women is an ascending infection of the uterine tube that is called salpingitis, inflammation of the fallopian tube, pelvic inflammatory disease, which can result in sterility or ectopic pregnancy as a result of scarring of the tubes. Disseminated gonococcal infection commonly manifest as arthritis, tenosynovitis, or pustules in the skin. Disseminated infection is the most common cause of septic arthritis in sexually active adults. The clinical diagnosis of the disseminated gonococcal infection is often difficult to confirm using laboratory tests because the organism is not cultured in more than 50% of cases. That's why it is difficult to diagnose. Other infected sites include the anorectal area, throat, and eyes. Anorectal infections occur chiefly in women. They are frequently asymptomatic, but a bloody or purulent discharge that is called proctitis can occur. In the throat, pharyngitis occurs, but many patients are asymptomatic. In newborn infants, purulent conjunctivitis ophthalmia neonatorum is the result of gonococcal infection acquired from the mother during passage through the birth canal. These are the different disseminated infections जिसमें एनोरेक्टल हो गया, pharyngitis हो गया और ophthalmia neonatum in the newborn हो गया when while passing through the birth canal. Neonatal conjunctivitis ophthalmia neonatum that is caused by the Neisseria gonorrhea during the passage through the birth canal. The incidence of gonococcal ophthalmia has declined greatly in recent years because of the widespread use of the prophylactic erythromycin eye ointment or silver nitrate applied shortly after birth. Gonococcal conjunctivitis also occurs in adults as a result of the transfer of organism from the genital to the eyes. Adult में भी conjunctivitis हो सकता है अगर genital से जो बैक्टीरिया है वो eyes तक ट्रांसफर हो जाता है. Other sexually transmitted infections like the syphilis and non-gonococcal urethritis caused by the chlamydia trachomatis can coexist with gonorrhea. Therefore, appropriate diagnostics and therapeutic measures must be taken. Along with the gonorrhea, syphilis and the chlamydia is also sexual transmitted disease. तो गोनोरिया के साथ साथ साइफल्स और क्लेमिडिया के जो सिम्टम्स वो भी अपीयर हो सकते हैं, इसलिए डिफरेंशियल डायग्नोसिस की इम्पोर्टेंट ज़रूरत है. दैट्स व्हाई वी टेक द इम्पोर्टेंट एंड द एप्रोप्रिएट थेरेप्यूटिक डोज टू ट्रीट gonorrhea, either it is a gonorrhea, syphilis or the chlamydia. जो भी उसके अगेंस्ट हम स्पेसिफिक therapeutic dose इस्तेमाल therapeutic measures will be taken against their specific infectious agent. Laboratory diagnosis of the gonococcal infection, mainly Neisseria gonorrhea, the specimen that are required for the lab diagnosis are the pus and secretion are taken from the urethra in case of male, cervix, rectum, conjunctiva, throat, or synovial fluid for culture and smear. Blood culture is necessary in systemic illness, but a special culture system is helpful because gonococci and meningococci may be susceptible to the polyethylene sulfonate present in the standard blood culture media. The diagnosis of urogenital infection depends on gram staining and culture of the discharge. However, nucleic acid amplification tests are widely used as a screening test. Nucleic acid amplification tests can be used. In men, the finding of gram-negative diplococci within the polymorphoneutrophils in a urethral discharge specimen is sufficient for diagnosis. In women, the use of gram stain alone can be difficult to interpret. Therefore, culture should be done. अगर मैन में gram-negative diplococci polymorphoneutrophils में आइडेंटिफाई हुए, it means it's a Neisseria gonorrhea. लेकिन फीमेल में ग्राम स्टैनिंग पे टोटली रिलाई नहीं किया जा सकता. Gram stains on cervical specimens can be falsely positive. because of the presence of gram-negative diplococci in the normal flora and can be falsely negative because of the inability to see small numbers of gonococci when using the oil immersion lens. ये false positive भी हो सकता है और false negative भी हो सकता है. false positive क्यों हो सकता है कि जो ना ग्राम नेगेटिव बैक्टीरिया हैं जो डिप्लोकोकाई हैं जो नॉर्मल फ्लोरा के तौर पर काम कर रहे हैं वो एज अ डिप्लोकोकाई आइडेंटिफाई होंगे सर्विकल स्पेसिमेंस में दैट्स व्हाई इट मे इंडिकेट एज अ फॉल्स पॉजिटिव फॉल्स नेगेटिव कैसे होगा एक्चुअली ये तो होगा पॉजिटिव में फॉल्स नेगेटिव के बाज़ औक़ात वो देखने में मसला होता है प्रॉब्लम होता है the gonococci are unable to see while using the oil immersion lens, so that's why it is a false negative. It may be false negative. Culture must be used in diagnosing suspected pharyngitis or anorectal infections. Specimens from mucosal sites such as the urethra and cervix are cultured on thyre morton medium, which is a chocolate agar containing antibiotics, vancomycin, colistin, trimethoprim, and the Neisseriae to suppress the normal flora. थायर मॉडर्न मीडियम में जो एंटीबायोटिक्स हम यूज करते हैं दैट आर यूज्ड टू इनहिबिट to suppress the normal flora to grow specifically Neisseria gonorrhea. Note that specimen from sterile sites such as the blood or joint fluid can be cultured on chocolate agar without antibiotics because there is no competing normal flora. क्योंकि जो ब्लड हैं जो इंटरनल हमारे पास जॉइंट फ्लूइड हैं या सी एस एफ है सेरेब्रोस्पाइनल फ्लूइड है उसको जब कल्चर करेंगे उसको थायर मैटर मीडियम पे कल्चर करने की ज़रूरत नहीं है उसमें एंटीबायोटिक ऐड करने की ज़रूरत नहीं है उसको हम डायरेक्ट चॉकलेट एगर पर भी ग्रो कर सकते हैं बिकॉज द नॉर्मल फ्लोरा is not mainly present or found in the blood in the giant fluid or the cerebrospinal fluid. The finding of an oxygen-positive colony composed of gram-negative diplococci is sufficient to identify the isolate as a member of the genus Neisseria. चूंकि oxygen-positive है, तो जब oxygen-positive है, it means it is a Neisseria. Specific identification of the gonococcus can be made either by its fermentation of glucose but not maltose by the fluorescence antibody staining. चूंकि जो Neisseria Gonnorrhrae है, it can only ferment the glucose, not the maltose. जो मेनिंजाइटिस है वो ग्लूकोज और माल्टोज दोनों को फर्मेंट कर सकते हैं तो दोनों में डिफरेंस करने के लिए वी कैन यूज द ग्लूकोज फर्मेंटेशन अगर सिर्फ ग्लूकोज की फर्मेंटेशन होगी इट मीन इट्स द गोनोरिया अगर दोनों की फर्मेंटेशन होगी ग्लूकोज और माल्टोज की इट मीन इट इज द मेनिंजाइटिस न्यूक्लिक एसिड एम्प्लीफिकेशन टेस्ट ऑफन एब्रिविएटेड न्यूक्लिक एसिड एम्प्लीफिकेशन नेट डिटेक्ट द प्रेजेंस ऑफ गोनोकोकल न्यूक्लिक एसिड इन द पेशेंट स्पेसिमेंस Serology-based tests serum and genital fluid contain immunoglobulin, IgG, and IgA antibodies against gonococcal pili, outer membrane protein, and the lipopolysaccharide that is called lipooligosaccharide. Some IgM of human sera is bactericidal for gonococcal in vitro. In infected individuals, antibodies to gonococcal pili and outer membrane protein can be detected by immunoblotting, radioimmunoassay, and ELISA enzyme-linked immunosorbent assay. We can also use immunoblotting, radioimmunoassay, and the ELISA for the identification of the antibodies against the pili and the outer membrane proteins. However, these tests are not useful as diagnostic aids for several reasons, including gonococcal antigenic heterogeneity, the delay in the development of antibodies in acute infections, and a high background level of the antibodies in the sexually active population. These are different drawbacks while using the serological detection. Note that serological tests to determine the presence of antibody to gonococci in the patient serum are not useful for diagnosis because of this antigenic heterogeneity. Treatment of the gonococcal infection. Ceftriaxone is the treatment of choice in uncomplicated gonococcal infections. Ceftriaxone, that is the third generation cephalosporin. If the patient is allergic to penicillin or the cephalosporin, a regimen such as the gentamicin plus azithromycin can be used. अगर patient allergic हो penicillin का cephalosporin का, then we can use gentamicin along with the azithromycin. Because mixed infection with chlamydia trachomatis are common, azithromycin or doxycycline should be prescribed in addition to ceftriaxone. तो sometime जो gonorrhea है उसके साथ chlamydia trachomatis का infection, chlamydia trachomatis का इन्फेक्शन भी हो सकता है, इसलिए एजिथ्रोमाइसिन के साथ एजिथ्रोमाइसिन को भी इस्तेमाल कर सकते हैं इन एडिशन टू सेफ्ट्राइक्सोन. A follow-up culture should be performed 1 week after completion of the treatment to determine whether gonococci are still present. Treatment of complicated gonococcal infection such as the pelvic inflammatory disease typically required hospitalization. Prior to the mid-1950s, all gonococci were highly sensitive to penicillin. Subsequently, isolates emerged with low-level resistance to penicillin and to other antibiotics such as the tetracycline and chloramphenicol. This type of resistance is encoded by bacterial chromosome and is due to reduced uptake of the drug or to altered binding sites rather than the enzymatic degradation of the drug. Then in 1976, penicillinase producing strains PPNG, penicillinase producing Neisseria Gonnorrhrae strains that exhibited high level resistance were isolated from patients. Penicillinase is plasmid encoded. Isolates resistance to fluoroquinolones such as the ciprofloxacin have become a significant problem and fluoroquinolones are not recommended as treatment. Isolate resistance to sulfonamide and the tetracycline have also been recovered. In 2017, the WHO reported that several strains of gonococci resistance to all antibiotics have been isolated. शुरू में कोई भी इन्फेक्शन नहीं था, कोई भी ऐसा ऑर्गेनिज्म नहीं था, बैक्टीरिया नहीं था जो नाइजीरिया गोनोरिया, ऐसा कोई नहीं था जो रेजिस्टेंस हो, लेकिन 2017 में ऐसे स्ट्रेन्स गोनोकोकाई के आइसोलेट हुए, रिपोर्ट हुए जो तमाम टाइप ऑफ एंटीबायोटिक से रेजिस्टेंस हैं. Prevention of the gonorrhea infection, gonococcal infection. The prevention of the Neisseria gonorrhea involves the use of PPE, personal protective equipments, and the prompt treatment of the symptomatic patients and their sex partners. Cases of gonorrhea must be reported to the public health department to ensure proper follow-up and contact tracing. केसेस को हाइड नहीं करना चाहिए, उनको रिपोर्ट करना चाहिए ताकि उनकी प्रॉपर फॉलोअप हो और प्रॉपर उसकी ट्रीटमेंट हो. ए मेजर प्रॉब्लम इज द डिटेक्शन ऑफ एसिम्प्टोमेटिक कैरियर्स. जो लोग एसिम्प्टोमेटिक कैरियर होते हैं उनको डिटेक्ट करने में प्रॉब्लम होता है क्योंकि उनकी डायग्नोस्टिक्स सही नहीं हो रही होती, उनको आइडेंटिफाई नहीं किया जा रहा होता, उनमें सिम्टम्स अपीयर नहीं होते. दैट में spread the infection to other peoples. Gonococcal conjunctivitis in newborns is prevented most often by the use of erythromycin ointment or silver nitrate ointment. Silver nitrate drops are used in some countries. No vaccine is available for the Neisseria gonorrhea because of the antigenic heterogeneity and the phase variation of the Neisseria gonorrhea. That's all about the gram-negative cocci Neisseria gonorrhea. Mainly infect the genitourinary tract, STD, sexual transmitted infection, having the different types of the virulence factors. The virulence factor mainly having the pili, pore protein, upper protein, and the lipo-oligosaccharide. The most important character of the Neisseria gonorrhea is the phase variation and the antigenic variation. That's why it is unable to treat and it is unable to vaccinate or to produce vaccine against the Neisseria gonorrhea. The most important complication of the Neisseria gonorrhea is the Fitz-Hugh-Curtis syndrome. That is a rare disorder that mainly happens when pelvic inflammatory disease causes swelling of the tissue around the liver or the liver capsule. Fitz-Hugh-Curtis syndrome is the important complication of the Neisseria gonorrhea. Thank you so much.